Hi everyone. This is your fifth lecture for the chapter Neural Control and Coordination. In the last class, we completed anatomy portion of the chapter of your syllabus. Today, we will be talking about the nerve impulse conduction. Okay, how the signals are actually moving in your neurons. Okay, in your nervous system. So let us begin. Generation, which is how these impulses, these signals, from now on we shall be calling signals as impulses. Okay, so how are these signals or impulses generated? So they are generation and conduction. After being generated, how are they being conducted? That also we shall study in this class. In other words, this conduction of nerve impulse is also called action potential. Okay, but we shall see how there is a minor difference between this nerve impulse conduction and action potential. So let us start by talking about some basic things like excitable cells. Okay, from the name itself you can understand that these are the cells which can be excited. Okay, not all the cell of your body are excitable. Okay, only few cells like your neurons and your muscle fiber, they can be excited. Now what is this excitable cells? What does it mean? So these are the cells whose cell membranes are in polarized state. So wherever you hear this term polarized, poles, like north pole, south pole. Then in case of charges, you have positive end, negative end, right? Same way here also. Polarized means it has two different ends. Okay, two different charges. Okay, so for example, look at this diagram. Here you can see that there is a charge separation. Okay, across the membrane. So this membrane can be called polarized. What does it mean? In simple words, it means on one side there is negative charge and on the other side there is positive charge. Clear? So can you say this is positive pole, this is negative pole, right? So like this, polarized. Fine. So the cell membranes of your excitable cells are polarized. Why are they polarized? How are they polarized? So they are polarized due to differential concentration of ions across the membrane. Now focus here again. You can see that Na plus is present on the extracellular side, which is outside the cell. Okay. So the outside the cell is positive, and it is positive because there is a lot of sodium ions present outside. Talking about inside the cell, you can see there are a lot of potassium ions present, but along with these potassium ions, there are anions which are also present. Anions are negative ions. Okay, so because of these anions, you can see that inside the membrane you have negative charge. Okay, so this here is called your charge difference. As well as when you look at the concentration, then you can find that sodium is concentrated outside, there is very less sodium inside. So even there is a sodium ion concentration gradient. Okay, concentration is higher outside, concentration is lower inside. So you can see this is the kind of gradient which is formed. Then talking about potassium, potassium is higher inside and lower outside. Okay, now this was one of the questions asked in I think 2013 or 14 paper, that which ion is found in abundance or is found in higher amount inside a cell. So the answer is potassium. Okay, sodium is rarely required. The answer would be potassium here. We will again cover it in mineral nutrition, but here just I wanted to mention. Fine. So you can see that there is differential concentration gradient. Differential matlab there is a difference, right? The concentration is higher outside or higher inside, and vice versa. The charge difference across the exolemma. Exolemma is your cell membrane of axons. Okay, So cell membrane of exons, they also have such kind of charge separation. This is because of its selectively permeable nature. What does this mean, selectively permeable? It means that the cell membrane is very choosy. It will select only some things that can cross the membrane. Okay, not everything can cross the membrane. Remember, we have discussed such terms when we were studying cell. So cell membrane, we have discussed this term, selectively permeable. In case of mitochondrial inner membrane, also we have understood this term, selectively permeable means what. Fine. Now let us still discuss a little bit about the selectively permeable nature. So it will allow the ions to pass only through these ion channels, which is simple. Ions are charged particles, okay? So charged particles cannot move across this non-polar cell membrane. So they require a certain transporter or certain channels through which they can pass, okay? So depending on which channels are present, whether they are open or closed, the cell membrane will decide that what ions should come in, what ions should go out. Fine till here? Now let us discuss this cell membrane of your or exolemma, okay, cell membrane of your exon. Here we will be focusing first on the sodium channel, okay, which is shown by this color. As you can see, this is the sodium channel. These yellow colored balls outside are also sodium ions. Fine. So when the sodium ion channel is open, where will the sodium ions move? Then we saw that or we can even see in this diagram that sodium concentration is higher outside and sodium concentration is lower inside. So the charges, which in this case is sodium, it will move from its higher concentration to its lower concentration. Clear? Second thing, when this channel opens, then you can see that outside is positively charged while inside is negatively charged. So what happens? To balance the negative charges, the positive ions, okay, here the sodium ions, they will move from outside to inside. Clear? So there are two reasons for the sodium to move inside. First reason is that its concentration is more outside and less inside. So it moves from its higher concentration to lower concentration. Okay, this is known as simple diffusion. And because you require a channel, so you can call it facilitated diffusion. Clear? Second thing, there is a charge difference. Okay, so the positive ions will move towards the negative side to balance out that negative charge. Clear till here? Okay. So here you can see a close-up view of these sodium channels. These sodium channels, which are involved in your nerve impulse conduction, are actually voltage gated sodium channels. Now, what does this mean? So, you may ask me that, Madam, when will this sodium ion channel open? Will it always remain open? If it always remains open, then all the sodium from outside will come inside till an equilibrium is reached, which is not possible. So, my answer to that is this. Okay, voltage gated channels मतलब कि जब voltage में change आएगा, only then these sodium ion channels will open. As you can see here, इधर से बंद कर दिया है. Okay, they have closed this sodium ion channel. Only when there is some voltage change, they will open up. Clear? So they are closed, and when there is some change in voltage, they will open. And as soon as they open, all the sodium ions, they will move inside. Okay, till the number of sodium ions outside and inside is same. Clear? Fine. So this is called equilibrium when the concentration of sodium outside and sodium inside is same. Fine. Chalo. Now let us look at the another channel, which is potassium ion channel, which is shown here. Okay. So this is potassium ion channel. you can see that the potassium ion, which is shown in this square or diamond shape, it is concentrated inside. Okay, so its intracellular concentration is high. These potassium ions will move from from inside the cell to outside. Clear? When they open. Same as sodium ion channels, these are also voltage gated potassium ion channels. Okay, from the name only, voltage gated, मतलब gate कौन खोलेगा? सिर्फ voltage खोल सकता है gate. When there is some change in the voltage, then only these channels will open and close. Fine. So when this potassium channel opens, the potassium will move outside the cell. Now the third structure that you see here, the third protein that you see, is called sodium potassium pump. Fine. Pump jahan bhi aata hai, wherever you hear the term pump, it means it is using ATP. Okay. The another name for sodium potassium pump is sodium potassium ATPase. Okay. It will use one ATP and do what? It is an anti-porter. Fine. Anti-porter matlab ek haath se le, ek haath se de. Okay. It will take some ions from inside to outside, and at the same time it will bring some ions from outside to inside. Clear? So this two-way exchange will occur through the sodium potassium pump. Fine. As you can see, it will pump these sodium ions, 3 sodium ions out, and it will bring 2 potassium ions in. So that is the function of sodium potassium pump. There are one or two few more channels. For example, voltage gated calcium ion channel. Okay, so there are some calcium ion channels which are present at the exon terminals. So when we discuss exon terminal, at that time we shall cover the calcium ion channels. Clear? But this much should be clear. These neurons and muscle cells, they are excitable. Excitable means that across their cell membrane, you have a charge difference. The outside is positive, the inside is negative. These charge differences are existing because the cell membrane is selectively permeable. What does it mean by selectively permeable? It has special proteins which can allow, okay, or disallow the movement of charges. Clear? So because of these different protein or ion channels, these charge differences are maintained. Chalo. Now let us actually begin with the nerve impulse conduction. Okay, before that, the location of ion channels. So if you want to know where exactly these ion channels are located, then in case of myelinated axon matlab which has this myelin sheath. Okay, in myelinated axons you find such ion channels. Wherever you hear, wherever you see this kind of structure, ये ion channel है और उसके साथ ऐसा ball like attached है. This is showing your sodium ion channel. Okay, and the other one is potassium ion channel. So you can see from here that these ion channels are present at the nodes of Ranvier. क्यों इधर ही present है, इधर कहीं पे नहीं दिख रहा? The answer is, remember, myelin sheath is an electrical insulator. It will not allow the charged particles to move outside and inside. Okay, so wherever this myelin sheath is present, the ion channels are not present in larger amount. Okay, they are mostly present in the nodes of Ranvier. Talking about unmyelinated or unmedullary axons, you can see that they have equal amount of ion channels present on the whole axon membrane. Clear? So this is the difference, or this is the exact place where you can find ion channels in myelinated and non-myelinated axon. Now beginning with the nerve impulse conduction. The normal phase, when the neuron is not doing any work, okay, it is at rest. So when neuron is not conducting impulse, it is known to be resting. Okay, you call that the neuron, okay, that neuron is resting. Fine. So this is called resting phase. The membrane is permeable to potassium but impermeable to sodium and anions. Okay, at resting stage, permeable to potassium मतलब potassium ion channels are open. कि अगर potassium को बाहर से अंदर आना है, then they can come like that. Okay, so they are permeable to potassium but impermeable to sodium. Sodium जो है वो बाहर से अंदर नहीं आ सकता, as well as anions. Now, if you remember the first diagram, on the inner side, okay, on the inner surface of your membrane, you have higher concentration of potassium. Okay, so the exoplasm, it has higher concentration of potassium as well as higher concentration of anionic proteins. So these are the proteins which have negative charges on them. My question for you would be, which all amino acids are present which can give the proteins negative charge? Okay, so anionic proteins are present inside, and there is less concentration of sodium on the inside, okay, in the exoplasm. In contrast, okay, the fluid outside the exon, which is the extracellular fluid, it has lower potassium but higher sodium. So because of this, there is a concentration gradient. Okay, let us look at the diagram now. You can see here the right hand side diagram. You have sodium more outside, less inside. So the triangle is like this. Okay, higher outside, lower inside. Then potassium is higher inside and lower outside the exoplasm. Okay, and anionic proteins, A minus, is also again excuse me, higher inside and lower outside. Because of this, the outside of the membrane is positively charged and the inside of the membrane is negatively charged. These ionic gradients are maintained by sodium potassium ATPase through active transport. Now let us understand these sodium potassium ATPases, fine, or which are also known as sodium potassium pump. What do? They take 3 sodium from inside. Okay, can you see the structure of this sodium potassium pump? They can bind to 3 sodiums, like here 1, 2 and 3. And on the other side, they can bind to 2 potassiums. Okay, let us understand step by step. So these 3 sodiums from inside, okay, which are present on the inside, they will bind here. There is a site where ATP will be lysed. Okay, so ATP will be broken to gain energy. With help of this energy, there will be some structural changes in the protein, okay, in the pump. From this conformation or this structure, it will now move and open like this. You can try it with your hands, okay. Here you can understand that the tips of your fingers are touching and your palms are away from each other. Now you pick up some sodium from inside, and now what you're doing, you're moving it in the opposite way, that your palms are touching and your fingers are free. So when this sort of movement happens, the sodium ions which were bound from the inner side, they are transported outside the cell membrane. Clear? So this is the way how sodiums are pumped out. Now along with that, the potassium which may be present outside, these potassium ions are picked up and the protein, okay, it regains its conformation. That means that again from palm touching, it will come back to the fingers touching and the palms being separate. Okay, and in this process, the potassium which has attached to the protein from outside, okay, it will now be released inside, and this is how you generate your membrane potential. Okay, or you are generating this difference in charge. बाहर थोड़े भी अगर पोटेशियम आयंस बचे कुचे जो भी पड़े हैं, you are bringing these potassium ions against their concentration gradient. याद रखो, concentration gradient मतलब अंदर ज्यादा है बाहर कम है potassium, but still यूजिंग एटीपी तुम क्या कर रहे हो, तुम धक्के मार मार के जितने भी पोटेशियम बाहर पड़े हैं सबको पिकअप कर करके अंदर की तरफ यू आर स्टफिंग देम. ओके, एंड सेम इज द केस विद सोडियम कि बाहर सोडियम ऑलरेडी हाई है बट फिर भी तुम पकड़ पकड़ के सोडियम को अंदर से खींच के बाहर की तरफ धकेल रहे हो. ओके, सो दिस इज द फंक्शन ऑफ दिस sodium potassium pump. Okay, it will push these ions against their concentration gradient. और अब तुमको समझ में आया कि why this gradient is maintained. Okay, why the sodium is high outside and potassium is high inside. ये maintain कैसे होता है. Fine. And this of course it requires energy. तुम खींच खींच के धकेल धकेल के उनके concentration gradient के against कुछ काम कर रहे हो तो तुमको energy लगेगी, and this energy you get from ATP. Yeah, so this is called active transport. Active transport मतलब transport कर रहे हो but you are using energy. Okay, now these sodium potassium pumps, they maintain these ionic gradients. The potential difference across the membrane at rest is called resting membrane potential. अभी तुम लोगों ने फिजिक्स में या कहीं पे पढ़ा होगा that what you can do, you can take two electrodes of let us say ammeter. Okay, no, let us take voltmeter, sorry. So you can take one from here and one from the inside, and there is this meter here which will show you the reading, right? जो voltage दिखाता है, voltmeter. So now what will happen, the outside is positive and the inside is negative. तो इन दोनों के बीच में जितना potential difference रहेगा that will be shown on your meter. This potential difference, okay, ये जो difference है, ये जो voltage है, that is known as resting membrane potential. Remember, potential और voltage, both of these things mean the same. दोनों का मतलब एक ही होता है. So this resting membrane potential, resting membrane क्योंकि अभी कोई signal आ नहीं रहा. Okay, there is no impulse. The neuron is at rest. तो ऐसे resting membrane potential जो है is around -70 millivolt. Okay, approximately -70 millivolt. माइनस का मतलब क्या होता है, इट जस्ट मीन्स दैट इनसाइड इज नेगेटिव, आउटसाइड इज पॉजिटिव. ओके, तो माइनस का मीनिंग याद रखना. वी वांट टू फाइंड आउट कि एक्सॉन के अंदर, न्यूरॉन के अंदर कितना है. तो दिस यू फाइंड आउट एंड द वोल्टेज विल शो माइनस. ओके, नेगेटिव साइन, एंड दैट नेगेटिव साइन मीन्स कि अंदर की तरफ यू हैव नेगेटिव चार्जेस. ओके, एंड रेस्टिंग स्टेज, and outside you have positive charges. Clear till here? I hope this much is clear to you. Fine, the resting membrane potential is around -70 millivolt. This number just remember. Fine. Okay, so this is at rest. Sodium bahar zyada, potassium andar zyada, and inside is comparatively negative, outside is comparatively positive. Kitna positive, kitna negative, then the voltage difference is -70 millivolt. Again, do not forget the unit, it is millivolt. Chalo. Now what happens? Chalo. अभी तुम एकदम रेस्ट में था न्यूरॉन, बट सडनली कोई सिग्नल आया या सिग्नल जनरेट हुआ. Okay, so once this depolarization occurs, what is this signal? This signal is nothing but it is depolarization. Okay, a nerve impulse or action potential is said to be generated. ये action potential या nerve impulse कैसे generate होता है? Then the generation will happen by depolarization. Polarization तुमको पता है, polarized मतलब there is a difference between the two charges. Okay, between the two sides, between the two ends. Depolarization मतलब अभी दोनों के बीच में difference नहीं रहेगा. Okay, that is what they try to tell from the name. So let us see what is happening. This happens, okay, this nerve impulse or action potential generation happens by opening of sodium ion channels. Clear? अभी ये रेस्टिंग मेम्ब्रेन पोटेंशियल था, यू कैन सी इट इनसाइड इज -70 मिलीवोल्ट, बाहर सोडियम ज्यादा अंदर पोटेशियम ज्यादा। अचानक से जब सिग्नल जनरेट करना है, इम्पल्स जनरेट करना है और एक्शन पोटेंशियल जनरेट करना है, all of these three things mean the same, then the sodium ion channels will open. ऐसे सोडियम आयन चैनल जैसे ही ओपन हुआ, what will happen? सोडियम तो बस ऐसे ही राह देख के ही खड़ा था कि चलो जैसे ही खुलेगा मैं सीधा अंदर घुस जाऊंगा. So that is what will happen. All of this sodium, okay, it will try and move inside towards its concentration gradient. Okay, बाहर ज्यादा है अंदर कम है तो बाहर से एकदम से अंदर आना शुरू हो जाएगा. This is called sodium influx. Okay, अंदर आना, when there is increased diffusion towards inner side, it is called influx. Because of this sodium influx, अभी exoplasm जो पहले negative हुआ करता था, look at this diagram, it used to be negative. Now because of this sodium, इतना ज़्यादा sodium आ गया कि अब exoplasm membrane, okay, towards your axon, okay, जो अंदर की तरफ है, exoplasm is cytoplasm of your axon. तो exoplasm की तरफ अभी positive charge दिखने लगा. Outside became comparatively negative. इतना ज़्यादा sodium अंदर आ गया कि बाहर की तरफ तुमको negative दिख रहा है. So can you see the difference between resting membrane and depolarization? There is change in this charge difference. Okay, पहले बाहर positive अंदर negative था, अभी depolarization के बाद और after this sodium ion diffusion, अभी बाहर negative हो गया अंदर positive हो गया. Clear? Okay. The membrane potential increases. Increases क्यों लिखा है? Because from minus it will be going up. Okay, it will be going towards positive side. It increases and becomes positive up to +30 or +45 millivolt. ये again from cell to cell, neuron to neuron differ करता है which type of cell it is, but around +30 +40 millivolt is normally considered. Just look here. ये ग्राफ को ध्यान से समझो. Okay, this I will only teach once, so pay all your attention here. On the y-axis you have voltage, potential difference है, voltage है in millivolt. On the x-axis you have time. Now here is what you are doing. सबसे पहले when there is resting membrane potential, okay, resting state, तभी क्या था voltage, it was -70. Clear? So this is your -70. Fine. Now something I want you to know is कि action potential जो है, it is an all or none phenomena. All or none मतलब या तो होगा या नहीं होगा. ऐसे आधा हो गया आधा नहीं हुआ ऐसा नहीं होगा. Okay, there is no partial way. Either it will happen or it will not happen. Either it is black or it is white. ऐसे. Clear? So अगर स्टिम्युलस जो है मतलब जो सिग्नल क्रिएट कर रहा है जिसकी वजह से सिग्नल जनरेट हो रहे हैं, if that stimulus is not strong, तो एक्शन पोटेंशियल जनरेट नहीं होगा. कैन यू सी? सो व्हाट डिसाइड्स, कौन ऐसा नक्की करता है, मतलब कौन ऐसा डिसाइड करता है कि कितना होना चाहिए लेवल, कि उसके पार गए तो प्लस और प्लस मतलब कि होगा, या किसके नीचे अगर रहे तो नहीं होगा. सो दिस लेवल इज कॉल्ड थ्रेशोल्ड. ओके, जैसे घर में तुम थ्रेशोल्ड बोलते हो ना, थ्रेशोल्ड मतलब जहाँ एक रूम खत्म होता है दूसरा रूम शुरू होता है, राइट? सेम वे हियर थ्रेशोल्ड. दिस थ्रेशोल्ड इज नॉर्मली -55 मिलीवोल्ट. ओके, -55 मिलीवोल्ट के आसपास होता है. अगर तुम्हारा स्टिम्युलस, तुम्हारा सिग्नल स्ट्रॉन्ग हुआ और ये -55 के पार चला गया तो डीपोलराइजेशन होगा. अगर ये -55 के पार नहीं जा सकता देन डीपोलराइजेशन नहीं होगा और इम्पल्स जनरेट नहीं होगा. क्लियर? सो हियर यू कैन सी दीज आर द फेल्ड इनिशिएशंस. मतलब स्टिम्युलस इतना स्ट्रॉन्ग नहीं था तो छोटा छोटा डीपोलराइजेशन होके खत्म हो गया. नर्व इम्पल्स जो है या एक्शन पोटेंशियल जो है वो जनरेट हुआ नहीं. क्लियर? फाइन. तो -70 जो रेस्टिंग स्टेट पे था, अभी कोई भी स्ट्रॉन्ग स्टिम्युलस आया, ऐसा समझो तुम्हारे फ्रेंड ने तुमको एक जोर जोर से ऐसे लगाई कान पे खींच के, राइट? तो इट इज अ वेरी स्ट्रॉन्ग स्टिमुलस। जैसे ही लगाया, तुमको फील हुआ कि लगाया गाल पे, राइट? सो व्हाट विल हैपन? फर्स्ट देयर विल बी डीपोलराइजेशन। डीपोलराइजेशन में अंदर की तरफ पॉजिटिविटी बढ़ रही है, तो वी आर मेजरिंग दैट ओनली। बिफोर इट यूज टू बी -70, नाउ इट विल गो अप टिल +30 या +40। क्लियर? चलो, सो दिस वाज ऑल अबाउट डीपोलराइजेशन। क्लियर? जहां पे तुम देख सकते हो दैट द वोल्टेज इज इंक्रीजिंग। व्हाई इज इट इंक्रीजिंग? सोडियम अंदर आ रहा है, सोडियम अंदर को एकदम पॉजिटिव बना रहा है। क्लियर? चलो, हाँ, बाय द वे दिस पीक, ये जो पीक जनरेट हो रही है, दिस इज नोन एज एक्शन पोटेंशियल। क्लियर? अभी देखते हैं क्या होता है। Depolarization of axon membrane causes potassium channel ions to open. ओके, जो तुम्हारी पोटेशियम आयन की चैनल्स है, उसका गेट ओपन हो जाएगा। फाइन, रिमेंबर वी हैड डिस्कस्ड कि वो वोल्टेज पे डिपेंड करता है। तो जैसे ही वोल्टेज माइनस से प्लस की तरफ मूव हुआ, पोटेशियम आयन चैनल्स विल ओपन देयर गेट्स। जैसे ही पोटेशियम आयन चैनल के गेट ओपन होते हैं, then your potassium ions will start moving outside, as you can see from this diagram. Clear? सब potassium बाहर move कर रहे हैं। Why? First thing, because potassium concentration is high inside, low outside, तो concentration gradient है। And second thing, बाहर की तरफ negative हो गया, अंदर की तरफ positive है, तो this positively charged ions will move outside, negative को balance out करने के लिए। Clear? Now because of this movement of potassium ion channel, potassium ions जितने full speed से potassium बाहर जा रहे हैं, at the same rate your resting membrane potential is restored. देखो, in this same graph you can see कि अभी repolarization हो रहा है। repolarization मतलब again your inside will become negative, outside will become positive. क्लियर? जैसे ही पोटेशियम आयंस बाहर जा रहे हैं, धीरे 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 फ्रॉम पॉजिटिव योर वोल्टेज इज कमिंग बैक टू नेगेटिव। कैन यू सी? तो वोल्टेज जो है वो पॉजिटिव से नेगेटिव जा रहा है। दिस मीन्स दैट इनसाइड और योर एक्सोप्लाज्म इज अगेन बिकमिंग नेगेटिव, एंड दिस इज कॉल्ड रीपोलराइजेशन। रीपोलराइजेशन होगा तो योर रेस्टिंग मेम्ब्रेन पोटेंशियल विच वाज -70 वो रिस्टोर होगा। क्लियर? सो दिस restoration occurs by opening wait a minute. uh, okay, this statement is not, it does not belong to repolarization, huh? This I think by mistake only I have put it here, but do not focus on that. Chalo, the membrane potential decreases. Okay, here you can see the membrane potential is decreasing and it becomes less than -70. Fine, so this is whole repolarization. Chalo, now let us understand how is the signal moving ahead. Okay, ये तो हमने समझ लिया। चलो, सोडियम आयन चैनल खुल रहा है, पोटेशियम आयन चैनल खुल रहा है, बट व्हाट इज हैपनिंग थ्रू दिस? हमारा सिग्नल कैसे आगे बढ़ रहा है? तो दिस नर्व इम्पल्स कंडक्शन will show you how the impulses are conducted ahead. Okay, तो ये impulse जो है, जो peak था, okay, that peak only is known as action potential. Okay, this peak here is called action potential. Fine, चलो, तो this region, now focus on this dark pink area which we are calling A. अभी A पे जैसे ही तुमको झापट लगाई एक जोर से, इधर सोडियम आयन चैनल्स ओपनड, सोडियम आयन चैनल खुल गए तो सोडियम अंदर आने लगा, अंदर आया तो इट बिकेम पॉजिटिव, तो योर ए सेक्शन इज नाउ पॉजिटिव। लेकिन ये तो सिर्फ इतनी जगह के ही सोडियम आयन चैनल खुला है, बाजू की जगह जो है, द नेक्स्ट रीजन ऑफ योर एक्सॉन, ओके, इट इज स्टिल नॉर्मल, वो अभी भी रेस्टिंग स्टेट में ही है। कैन यू सी? इट इज स्टिल नेगेटिव इनसाइड, पॉजिटिव आउटसाइड। सो व्हाट विल हैपन नाउ? then remember this from your chemistry as well as from your physics कि current जो है it moves from positive to negative. ठीक है, इतना याद रखना है, the current will move from positive to negative. So that is what is happening. अभी अंदर तुम देख सकते हो उधर positive है, and on the next region, okay, उसकी बाजू की जगह जो है on the same axon is negative. तो पॉजिटिव से नेगेटिव करंट जो है वो मूव होता है. क्लियर? सो द करंट विल मूव फ्रॉम पॉजिटिव टू नेगेटिव इनसाइड द एक्सॉन. और आउटसाइड अगर आप देखो तो आउटसाइड में पॉजिटिव, ये नेक्स्ट रीजन जिसका नाम आपने बी दिया है, तो बी पे पॉजिटिव है और ए अभी भी बाहर नेगेटिव है. तो व्हाट विल हैपन? फ्रॉम पॉजिटिव टू योर नेगेटिव द करंट विल फ्लो. तो ये एरोज जो है वो करंट की डायरेक्शन शो कर रहे हैं. ठीक है? तो फ्रॉम पॉजिटिव टू नेगेटिव इट मूव्स. तो अंदर अगर तुम देखो, इनसाइड इफ यू सी ऑफ एन एक्सॉन, तो जहां पे एक्शन पोटेंशियल जनरेट हुआ है उधर इनसाइड विल बी पॉजिटिव एंड द नेक्स्ट एरिया विल बी नेगेटिव. तो फ्रॉम पॉजिटिव टू नेगेटिव द करंट विल फ्लो. बाहर की तरफ तुम अगर देखो तो योर ए एरिया, ओके, जहाँ पे एक्शन पोटेंशियल हुआ उधर आउटसाइड इज नेगेटिव एंड द बी सेक्शन जो उसके बाजू का एरिया है दैट विल बी पॉजिटिव. सो फ्रॉम पॉजिटिव टू नेगेटिव द इम्पल्स विल मूव. क्लियर? हमको क्या चाहिए? हमको ये बाहर का करंट कोई मायने नहीं रखता. वी डू नॉट केयर अबाउट दिस आउटसाइड करंट. व्हाट इज इम्पोर्टेंट फॉर अस इज दिस इनसाइड करंट. हमको तो न्यूरॉन कैसे आगे बढ़ाता है वो देखना है ना, न्यूरॉन के बाहर जो भी हो रहा हो. फाइन. सो व्हाट इज हैपनिंग इन योर एक्सॉन, व्हाट इज हैपनिंग इन योर न्यूरॉन, दिस करंट इज मूविंग अहेड. अभी ये जैसे ही करंट आगे जाएगा तो एट योर बी, ओके, दिस बी जो लाइट पिंक में दिखाया है उधर also, there also, I mean, the action potential will begin. How does action potential take place? So there will be depolarization, there will be repolarization and achievement of resting membrane potential. ठीक है? तो इस तरीके से जो भी ये रीजन है, जो एक्शन पोटेंशियल है, वो आगे आगे बढ़ता जा रहा है. एंड दिस इज हाउ द नर्व इम्पल्स कंडक्शन टेक्स प्लेस. इसीलिए आई टोल्ड यू एक्शन पोटेंशियल और नर्व इम्पल्स कंडक्शन में थोड़ा बहुत डिफरेंस है. दिस एरिया, ओके, यू कैन से कि उधर एक्शन पोटेंशियल जनरेट हुआ, लेकिन हाउ इट इज ट्रैवलिंग फ्रॉम एरिया टू एरिया एंड द होल प्रोसेस टुगेदर, you call it as nerve impulse conduction. Clear? So this is how the nerve impulse conduction takes place. Chalo, a very brief summary of this nerve impulse conduction, या a very brief summary of the action potential. तुम्हारा resting membrane है, अभी rest पे है membrane. That means outside there is more of sodium, inside is more of potassium. Outside is positive, inside is negative. इसको बोलते हैं resting membrane potential, which is -70. अभी एक्शन पोटेंशियल जनरेट हुआ जिसका मतलब है डीपोलराइजेशन होगा. इन डीपोलराइजेशन द सोडियम आयन चैनल्स ओपन. व्हेन द सोडियम आयन चैनल्स ओपन, सोडियम मूव्स इन, ओके, and so there is depolarization. The membrane inside becomes positive, the membrane outside becomes negative. Okay, this is the meaning of depolarization. अभी डीपोलराइजेशन से यू आर डूइंग रीपोलराइजेशन. रीपोलराइजेशन मतलब तुम रेस्टिंग मेम्ब्रेन को रिकवर करने की कोशिश कर रहे हो. सो योर पोटेशियम आयन चैनल्स विल ओपन. दिस पोटेशियम विल मूव आउटसाइड. ओके, सो आउटसाइड अभी वापस पॉजिटिव, इनसाइड विल अगेन बिकम नेगेटिव. क्लियर? दिस इज रीपोलराइजेशन. अगर कोई पूछे आपको what is the difference between resting potential and repolarization if both of them are -70 millivolt? दोनों में क्या difference है? So then the difference is that in repolarization the concentration of potassium will be high outside, okay, and in resting potential the concentration of sodium is high outside. तो ये तो गड़बड़ हो गया. So to regain this resting potential you have sodium potassium potential, my mistake, sodium potassium ATPase. okay, ये क्या करेगा सोडियम पोटेशियम ATPase? ये जो गड़बड़ हो गई है कि अभी बाहर पोटेशियम ज्यादा है, अंदर सोडियम ज्यादा हो गया, that will be again undone. okay, तो अंदर से सोडियम को पकड़ पकड़ के बाहर और पोटेशियम को बाहर से पकड़ पकड़ के अंदर, and this you regain the resting potential. ठीक है? तो इस तरीके से resting membrane potential जो है या resting state जो है that is regained. So this is the whole summary of the nerve impulse conduction or action potential. इतना clear? चलो. Now there is a type of conduction known as saltatory conduction. Saltatory का मतलब होता है जंप करना, ओके, जंप कर करके जाता है. This kind of conduction takes place in myelinated fiber, मतलब जिसके ऊपर माइलिन शीट प्रेजेंट है उधर तुमको ये दिखेगा. In effect, the action potential jumps from node to node and passes along the myelinated axon. ये Ranvier's node से Ranvier's node तक jump करेगा and it is faster compared to non-myelinated axon. कितना fast है बताऊं आपको? This saltatory conduction, it occurs at the speed of 200 meter per second. तुम्हारी हाइट मीटर में देखो और फिर सोचो कि 200 मीटर पर सेकेंड मतलब कितना फास्ट हुआ. क्लियर? सो ये सॉल्टेटरी कंडक्शन जो है इट इज वेरी फास्ट, इट अकर्स इन माइलिनेटेड फाइबर्स. लेट अस सी नाउ. देखो ये नॉर्मल है, इधर तुमको दिख रहा है डीपोलराइजेशन हुआ है, ओके, और योर एक्शन पोटेंशियल हैज बीन जनरेटेड. अभी क्या होगा दैट बिकॉज दिस माइलिन इज प्रेजेंट, इट इज एन इंसुलेटर. जहां पे माइलिन होगा उधर करंट जो है वो पास नहीं हो सकता. ओके, सो वेयर विल द करंट फ्लो? अंदर तो माइलिन नहीं होता ना, एक्सॉन के अंदर देयर इज नथिंग. सो द करंट विल पास फ्रॉम दिस नोड, ओके, जहां पे पॉजिटिव है उधर से फ्रॉम पॉजिटिव टू नेगेटिव द करंट विल ट्रेवल. तो ये सीधा जाके नेक्स्ट रैनवियर्स नोड जो है उधर डीपोलराइजेशन करवाएगा. ओके, सो दिस इज व्हाट इज शोन हियर. फिर इधर से अगेन नेक्स्ट नोड पे डीपोलराइजेशन आपको दिखेगा. सो फ्रॉम नोड टू नोड ऐसा लगता है कि जंप करके जा रहा है. ओके, योर नर्व इंपल्स इज जंपिंग फ्रॉम दिस नोड टू दिस नोड. ओके, ये बीच में कहीं पे भी नहीं होगा. सो दिस इज कॉल्ड saltatory conduction. It is way faster, okay, because ये सीधा जंप कर करके जा रहा है तो it is very fast compared to your non-myelinated axon. Let us look at it in this video form. Okay, you can see कि ये सीधा red color जो है वो show कर रहा है depolarization. So there is depolarization at one node, directly pass to the next node and the third node, and so it is moving fast. Okay, नीचे तुमको ग्राफ में भी दिख रहा है peaks जो है they are showing you the action potential at different nodes. ठीक है, अभी इसको ही कंपेयर करते हैं विथ नॉन माइलिनेटेड फाइबर. नॉन माइलिनेटेड फाइबर में सॉल्टेटरी कंडक्शन नहीं होगा. सो व्हाट विल हैपन, द रीजन, ओके, जो बाजू बाजू का रीजन है सभी जगह पे आपको एक्शन पोटेंशियल दिखेगा. सो इट विल मूव स्लोली, एकदम धीरे से मूव होगा, एंड एवरी पार्ट ऑफ योर एक्सॉन विल गेट depolarized, repolarized, and resting membrane potential recovered. ओके, तो हर एक पार्ट जो है वो इन्वॉल्व है. वाइल इन दिस केस सॉल्टेटरी कंडक्शन में तुम देख सकते हो ओनली द नर्व्स आर द नोड्स आर इंक्लूडेड इन कंडक्शन ऑफ द नर्व इम्पल्स. बाकी जहाँ पे माइलिन शीथ जो है प्रेजेंट है उधर नर्व इम्पल्स कंडक्शन या एक्शन पोटेंशियल नहीं दिखता. या सो दिस इज द डिफरेंस. एंड इवन हियर यू कैन सी कि जो फर्स्ट है वो एकदम फास्ट हो रहा है एंड सेकेंड वन इज कंपेरेटिवली स्लो. देख सकते हो. Okay. Now this we will start from the next class, which is अभी हमने ये तो देखा चलो कि किस तरीके से nerve impulse generate होता है. Okay, how is it starting? We saw how it is getting conducted. आगे कैसे जाता है. Okay, on your axon only. Next, in the next class we will see कि ये एक न्यूरॉन से दूसरे न्यूरॉन तक कैसे जाता है, which is known as transmission of impulses. Okay, इसको impulse conduction नहीं, transmission बोलते हैं. So transmission of impulses we will study from the next class. This will include this conduction through synapses. We will study types of synapses. We will also study types of neurotransmitters. Okay, and along with that we shall cover reflex arc, and if time permits we shall start with the sensory organs. Okay, so by end of this week easily, or within let us say 3 lectures at least, we may be able to complete this lecture, this chapter. So see you next time.